Hello, I'm Parham Arabzadeh. I'm 19 years old. I graduated from Allame School and obtained a diploma. The title of my project is Study of Molecular Pathways Involved in Bipolar Disorder. The achievements of my article are as follows: ICPC Tournament Gold 2024 from Korea and LTE Tournament Gold 2023 from the UK. Among the reasons I chose this topic are the increase in the incidence of this disorder in recent years, the high suicide rates among these patients, and the cardiovascular, metabolic, and endocrine disorders that stem from this disease. Our priority in this paper is to explore the molecular pathways involved in bipolar disorder. Through our research on the genes associated with bipolar disorder, we aim to develop effective treatments and facilitate the early identification of patients. As you can see, here are the number of articles retrieved in bipolar disorder. Bipolar disorder is a mood disorder resulting from genetic and environmental factors. According to the World Health Organization, bipolar disorder is the sixth most common medical disability among individuals aged 15 to 44. Adolescents are particularly vulnerable to the onset of bipolar disorder. According to the recent studies, mRNAs control key functions and processes such as nerve growth, synaptic development, plasticity, and circadian rhythm. Therefore, any disorders in these functions can cause psychiatric disorders. Blood samples from 95 hospitalized bipolar disorder patients were taken during both manic and euphomic phases. The Young Mania rating scale was used to assess individuals' manic phases. There were no significant differences in the prescribed medications between mood phases. In this project, the maximum possible number of male patients was examined. For this purpose, differential gene expression was used and optimized by comparing two groups. Patients with the greatest difference in the Young Mania rating scale scores between manic and euphomic phases were selected. Data analysis was conducted using SAS 9.2. Through bioinformatics analysis, 10 genes were identified as potentially involved in the development of bipolar disorder. These genes are implicated in various biological processes, signaling pathways, and functional annotations. Dysregulation of ion transport, neural development, and neurotransmitter signaling pathways may contribute to the development of bipolar disorder. Further research is needed to elucidate the precise mechanisms by which these genes influence the development of bipolar disorder and to develop more effective treatments. Continued studies are essential to identify the exact mechanisms through which these genes affect bipolar disorder, which may ultimately lead to the discovery of the novel drug targets and therapies. This project has allowed us to identify several genes associated with bipolar disorder and develop a diagnostic kit for detecting these genes. This tool enables the identification of individuals at high risk for the disorder before they exhibit symptoms, facilitating early monitoring and management. Early intervention can prevent the disorder from worsening and reduce the risk of relapse. Additionally, the identification of these genes may prompt further research into their potential therapeutic applications.